Hello, welcome to Biology for You. In this video, we are going to discuss nanopore DNA sequencing. In this series of videos, we have been discussing different DNA sequencing technologies. So, as a part of one of the DNA sequencing technique, we are going to discuss this nanopore DNA sequencing technique. So in this video we are going to have the basic idea of nanopore DNA sequencing. That means what exactly the nanopore DNA sequencing is and how it works, and followed by what are the advantages with it. So coming to the first point, what exactly nanopore DNA sequencing is. Basically, nanopore DNA sequencing is considered to be third generation DNA sequencing technique. That means it is very much efficient and also cost effective and time saving process. The other advantage with nanopore DNA sequencing is it is actually the machinery is very much simplified, unlike the other DNA sequencing techniques. So the machinery is very much simplified, that it is just a portable machine in which the DNA sample can be taken and it will sequence and gives the entire sequence of the DNA out of it. And the foremost important advantage with the nanopore DNA sequencing is it is basically need not to go for here in this nanopore DNA sequencing, we need not to go for clonal propagation, or maybe we need not to go for PCR amplification as well. That means a DNA molecule, a genomic DNA molecule, a linear form of DNA can be directly taken for the sequencing and without any kind of modifications. As such, the DNA sequencing will be carried. That means we need not to go for cloning into vectors, prepare a genomic DNA library, that is also not needed, and even we need not to fragment it and we need not to go for PCR amplification, and all these tedious processes are omitted in the case of nanopore DNA sequencing. So the DNA, double-stranded DNA or RNAs can be directly taken, and that will be directly sequenced. So in a single stretch of time, the DNA sequencing will be happen, and it is not depend upon the size of the DNA. So any length of the DNA can be simply sequenced. That means any genomic DNA from the complex organisms can also be sequenced in a single stretch of experiment. So by using it, genomic DNA can be sequenced without any complications, with the easy process. And these are the basic advantages with the nanopore DNA sequencing. So once after we understood the advantages, first we need to know how all these are possible, because with the next generation DNA sequences also, those next generation or NGS methods are also considered to be efficient, cost effective and also time saving. But this nanopore DNA sequencing is much advanced than those in terms of time, because there is no processing of the sample itself, and also a single stretch of DNA, that is what all required for the nanopore DNA sequencing. So going into the detailed process of the nanopore DNA sequencing, basically nanopore DNA sequencing deals with a nanopore. This nanopore is generally in nature, there are many membrane pores present on the plasma membranes. These membrane pores are helpful for the membrane to allow transport across the membrane, maybe from outside to inside, maybe from inside to outside. So these are very much essential for the survivability of the cell. That means the communication between the living cell and the environment is through this pores. So any kind of signal molecules, any kind of ions, maybe small molecules, all will come across the membrane through these nanopores. So these are basically membrane units. That means these are protein units embedded inside the plasma membrane. So they make physical barriers that allow the typical size of molecules to go through it. So these type of membrane proteins will be directly taken for the nanopore DNA sequencing, or maybe synthetically they are prepared using polymers. So such membrane pores or the nanopores, which are very much small, are embedded in the electrically resistant membrane. So during the DNA sequencing, first we need to isolate the DNA in its pure form, and we all need to do is just we need to allow this DNA to passes through this nanopore, which is fitted on the electrically resistant membrane. So here this DNA will be tagged with the one processing protein, and this processing protein will allow the DNA end of DNA to find this nanopore, and it will find the nanopore and sit upon it. So this processing protein has generally have a helicase property. That means it unwinds the DNA and allow the one strand to pass through the nanopore. So during the process of DNA sequencing, the first DNA is isolated, and that will be fused with the at one end. The DNA is fused with the processing protein, and that processing protein will allow the DNA to find the nanopore, and this processing protein will also come with this DNA, and also it is having helicase property to unwind the DNA, and it also allow one strand of DNA to pass through this nanopore. So that is the process of the nanopore DNA sequencing. So basically, as we already discussed, this is electrically stable membrane, and it is fused with the nanopore, and the electrical current is applied across the membrane. So due to the electrical field application, the ions will be okay, let us draw like this. Small ions will be passes through this membrane. This electrical current, so this type of ions will pass across the membrane when the electrical field is applied across it. So these ions will cross the membrane through this nanopore. This nanopore is basically a protein unit which has a halo structure inside it, which makes the barrel kind of structure, and through this empty area the ions will be easily passes through. So this electrical field will be disturbed once after this DNA is started passing through it. That means these ions, the flow of ions will be disturbed. So this flow of ions will be disturbed due to the passes of the DNA across it. Basically, if the diameter of the DNA is also very much suitable for the passing it through this membrane protein. Otherwise, if any protein molecule is allowed to passes through it, it cannot simply allow the passes of the larger molecular weight components such as proteins, because the diameter of the the diameter of the nanopore is such small. So it is generally up to 1 to 1.4 nanometers only. So the only single-stranded DNA can be passes through it. When this single-stranded DNA will be passing through this nanopore, the ionic current will be disturbed. But it is not the disturbance is not static. It will change, depends upon the type of nucleotide present on it. So this new DNA is nothing but a polymer of nucleotides. There are 4 kinds of nucleotide: A, T, G and C. So when the DNA will be passing through this membrane, there will be actually some holder type of structures which really holds the DNA for certain time when it passes across the membrane. So the DNA during that time, very small period of time, when it simply passes, one nucleotide will simply passes through the will pass across the membrane, the ionic current will be disturbed and the flow of ions will be disturbed due to the physical obstruction. So that will be that electrical disturbance will be monitored by the detector system. So depends upon the type of nucleotide that has been passing through the membrane, the ionic current change will be different, that will be measured by the detector system. So there will be such graph. That means the flow of ions or the electrical current will be disturbed if a nucleotide, adenosine nucleotide passes through the membrane. The electrical field, the change obtained in the electrical field will be different, and if guanine nucleotide is passing through it, then the electrical current change will be different. So it forms a peak similar to this. That means there are 4 levels of peaks that are possible due to the passes through the membrane. So one is for adenine, second one is for guanine, and maybe one is for cytosine and thymine. So based on that, every nucleotide that has been passing through the membrane will be detected. So this type of graph will be generated and that will be used to read the sequence of nucleotides present on the DNA. So that is how the nanopore DNA sequencing will measure or detect the order of nucleotides present on the DNA molecule. So once after this single strand is completely unwound and the other strand generally, so there will be 2 strands because the DNA is generally double stranded in nature. So once after the whole single stranded DNA had been passes through this membrane pore, the other strand will also be passes through it. That means there will be a structure which clips these 2 strands together. So that allow the once after one strand of DNA will be passing through, the other strand will also be allowed to passes through the membrane. That means for every single DNA molecule, at least 2 sequences will be obtained. One is from one strand of DNA and other is from its complementary strand of DNA. So it is very much efficient, cost effective as well as time consuming, and the only requirement is this one such system which is having one membrane pore, fusion protein, processing protein and a electrical membrane and some electrical field that could be applied to it along with the DNA that is going to be sequenced. So that is how nanopore DNA sequencing will be helpful to understand the sequence or order of the nucleotides present on the DNA.